All right, today we're diving deep into the very instruction manual for life itself, the central dogma of molecular biology. Now, I know that sounds a bit textbooky, but trust me, this isn't just abstract science. This is the core process that explains everything, from how our bodies work perfectly to the molecular train wrecks that cause cancer, genetic disorders, and even viral infections. We're going to follow the information from the master blueprint in our DNA all the way to the proteins that do all the work, and see how cracking this code is the absolute key to modern medicine. So, here's our game plan. First, we'll hit the main principle, the central dogma. Then we'll follow the code as it gets copied in replication, transcribed into a message, and then translated into a final product. And to wrap it all up, we'll connect everything back to what it actually means in the clinic. Let's jump in. Okay, first up, life's central dogma. This is it. This is the fundamental rulebook that explains how the instructions for building and running a living thing are read and used in, well, basically every cell in your body. What's so beautiful about the central dogma is just how logical it is. The information flow is a one-way street. Think of it like this. Your DNA is the master blueprint, locked away safely in the nucleus for protection. You can't take it out. So you make a photocopy. That's your RNA message. That mobile copy then leaves the nucleus and goes to the construction site in the cytoplasm, where workers, called ribosomes, read it to build the final product, the proteins. And it's the proteins that do, well, pretty much everything. So let's get into that first step. Before any cell can divide, whether it's for growth, to heal a cut, or unfortunately to form a tumor, it has to make a perfect copy of its entire library of instructions. That process is DNA replication. It's absolutely essential. Now, clinically, this is huge. This absolute dependency on replication is a major vulnerability we can target. What defines cancer cells? Uncontrolled rapid division. That means their replication machinery is always running on overdrive, and that's their weak spot. Many of our classic chemotherapy drugs are designed to sabotage this exact process. Some, like 5-FU, are basically fraudulent building blocks. The cancer cell tries to use them to build new DNA, and it just jams the whole machine. Others, like cisplatin, are more direct. They just go in and damage the DNA template so badly that the cell has no choice but to trigger self-destruct. We're essentially throwing a wrench into the engine of these rogue cells. The accuracy of this whole process is just mind-blowing. The main enzyme doing the building, DNA polymerase, is pretty good on its own, but it still makes mistakes. That's where the cell's quality control team comes in. These incredible proofreading systems constantly scan the newly made DNA, find the typos, and fix them. They take the error rate from about 1 in 100,000 down to a staggering 1 in a billion. That's an incredible level of precision. The whole thing works like a beautifully coordinated molecular machine. First, you've got an enzyme called helicase. Its job is to act like a zipper and just unwind the DNA double helix. But the main builder, DNA polymerase, can't just start on its own. It needs a starting line. So an enzyme called primase comes in and lays down a little RNA primer. Think of it as the starting block. Then, the star of the show, DNA polymerase, gets to work building the new strand. And finally, once all the pieces are there, DNA ligase comes in like a welder to seal up all the gaps, leaving you with 2 perfect DNA molecules. Okay, now here's a little quirk that makes things interesting. DNA has direction. We call it 5' and 3'. And the key rule is that DNA polymerase is a one-way street. It can only add new pieces to the 3' end. So for one of the DNA strands, the leading strand, it's smooth sailing. The polymerase just zips along, making one continuous new piece. But the other strand, the lagging strand, is facing the wrong way. So the polymerase has to work backwards, building it in a bunch of short little segments called Okazaki fragments, which then have to get stitched together later. It's a bit more complicated, but it gets the job done. So what happens when that amazing quality control system we just talked about fails? Well, the fallout is devastating. We see some pretty serious genetic diseases. If the system that fixes UV damage from the sun, nucleotide excision repair, is broken, you get xeroderma pigmentosum, and patients are at extreme risk for skin cancer. If the mismatch repair system fails, like in Lynch syndrome, little errors from replication build up, especially in tissues that divide a lot, like the colon, leading to a huge risk of colorectal cancer. And of course, you've heard of BRCA1. When that gene, which is critical for fixing major DNA breaks, is mutated, it dramatically increases a person's lifetime risk of breast and ovarian cancer. All right, on to our next step, transcription. So we've got our perfect master copies of DNA. Now the cell needs to read a specific recipe from the cookbook without risking damage to the whole book. So it makes a temporary working copy of just one gene. This process is called transcription. So how does a cell know which gene to turn on and when? It all starts with a signal. Special proteins called transcription factors get the message, and they go and bind to a specific spot at the beginning of a gene called the promoter. You can think of this as a big flashing start here sign. That sign then recruits the main enzyme RNA polymerase, which latches on and starts making that RNA copy of the gene. But that first RNA copy isn't ready for prime time. It's like a rough draft. It needs some serious editing. First, it gets a protective helmet, called a 5' cap on one end, and a long stabilizing tail, the poly A tail, on the other. But the most important edit is called splicing. The initial copy has a bunch of junk segments, called introns, that don't code for anything. Splicing is the process of cutting all of that junk out and stitching the important coding parts, the exons, together to create the final, clean message. And this editing process leads to something that is just one of the most elegant and efficient tricks in all of biology: alternative splicing. See, by choosing which exons to stitch together, a cell can create many different versions of a protein from the exact same gene. A liver cell might splice the message one way to make protein A, while a brain cell uses the very same gene but splices it differently to make protein B. This is a huge reason why we're so complex without needing a ridiculous number of genes. It's all about mixing and matching. Back in the clinic, this is where personalized medicine really comes to life. We can now analyze a patient's tumor and look at its gene signature. Basically, we see which genes are being transcribed or turned on. This gives us a detailed blueprint of what that specific cancer is doing. For example, if we see a breast cancer tumor is overexpressing the gene for a protein called HER2, we know that a targeted drug like Herceptin, which blocks HER2, is very likely to work. We're not just treating breast cancer anymore. We're treating that patient's specific cancer based on its RNA messages. Okay, so the edited message, the mRNA, has left the nucleus. It's time for the final act. It's time to actually build something. This is translation, the process of decoding that message and turning the language of genes into the language of proteins. So how does the cell read the message? It's simple, actually. The genetic code is read in 3-letter words. We call these codons. The ribosome, the cell's protein factory, moves along the mRNA, reading it 3 letters at a time, and each 3-letter codon specifies one of the 20 amino acids that needs to be added to the growing protein chain. The whole assembly line works in 3 phases. First is initiation. The ribosome finds the official start signal on the mRNA. Then comes elongation. The ribosome chugs along the message, reading codon after codon. For each codon it reads, a special molecule called a transfer RNA, or tRNA, acts like a delivery truck, bringing in the one specific amino acid that matches that codon. And finally, termination. The ribosome hits a stop codon, the assembly line halts, and the newly built protein is released. But wait, we're not quite done. A straight chain of amino acids isn't a functional protein. It's just a string. Function is all about the final 3D shape. So that string gets sent to the cell's origami masters, the ER and the Golgi, where it gets folded and modified into its precise, final, functional shape. And this folding is critical. If it misfolds, it can lead to all sorts of diseases, from cystic fibrosis to Alzheimer's. The final shape is everything. All right, let's tie it all together, from the code to the clinic. We've gone through this whole molecular journey, so why does it matter so much to a future doctor, a researcher, or anyone in healthcare? This quote just nails it. At its heart, modern medicine is just applied molecular biology. You can't fix a broken engine if you don't know how it's supposed to work in the first place, right? The same is true for the human body. By deeply understanding this normal pathway from DNA to protein, we can pinpoint exactly where things go wrong in a disease. And then we can start designing smart ways to fix it. So, if you remember one thing, let it be this. Pretty much every disease has a molecular root cause. Whether it's a typo in the DNA code, a splicing error that messes up the RNA message, or a final protein that gets misfolded, the problem starts somewhere along this fundamental pathway. This is the real battlefield for human health. And that leads us to the future, which honestly is already here. We can now sequence a person's entire genome. We can analyze their RNA. We have direct access to their personal biological code. So the final thought I want to leave you with isn't about the science itself, it's about what you're going to do with it. As you move forward in your careers, how will you use this incredible power, the power to read a patient's genetic code, to fundamentally change the way you practice medicine?